Good evening friends, welcome to my channel. So today we are going to discuss about histology spotters. So as you all know that histology, it is playing a major role, and also it is a bit complicated in the practical examinations. In order to find the slides, it is very difficult. So many slides are similar. So today's class, what we are going to do is how to identify the slides. So based on the steps, based on the points, we can suspect which slides they might be. Okay, so let's get started. Before that, please subscribe to my channel, those who have not done it. Okay, so the first and foremost slide which we are seeing on the screen is the hyaline cartilage. How to identify this hyaline cartilage? So hyaline cartilage identification is, you can see this is the entire, this matrix is called as homogeneous matrix, homogeneous basophilic matrix. Basophilic means it appears blue in color. Okay, eosinophilic means pink in color. So this hyaline cartilage, it appears blue in color. Okay, so this basophilic matrix, it appears blue in color, and these are called as chondrocytes. They are called as cartilage cells. So they looks like eyes. Okay, so when you are looking into the microscope, so the eyes, they just appears like the eyes. They are looking at your eye. Okay, that is an easy identification of this hyaline cartilage, the chondrocytes. Okay, so other features what you have, you might have studied about it. Simple identification is, it is lined by the pericardium. Then the matrix, it appears blue in color, and the chondrocytes, they appears like the eye. Okay, and the chondrocytes, they are present in groups. So the groups are called as cellness. So what you have to write in the second question, first you will identify the slide that it is a hyaline cartilage. The second identification points what you have to write is, so appear presence of the chondrocytes in a groups, then homogeneous basophilic matrix is seen, like that you can write. The next, we are moving to next slide, and this is how we can identify in the slide. Okay, so perhaps you may not see clearly that basophilic matrix, but you can identify the groups of chondrocytes called as cell nest. Okay, that is about the identification of hyaline cartilage. So this slide is white fibrous cartilage. How to identify the white fibrous cartilage is, so actually these diagrams which I have taken from the D4 Atlas, D4 Atlas, and also the Balakrishna Shetty textbook, and some hand-drawn diagrams also I have taken. So just to make you comfortable and make you easy to find the slides. So this white fibrous cartilage identification is, so entire the thing is, it appears like the fibrous part. Okay, all the fibers you can see, and occasional chondrocytes you can identify. It means the less number of chondrocytes are present, and they are present in the rows. Okay, they are present in the rows. So identification of the white fibrous cartilage is, there is no pericardium. Can you see any pericardium? No. So there is no pericardium, and also the chondrocytes are arranged in rows. Like that, you have to identify the white fibrous cartilage. Yes, coming to the white fibrous cartilage, this is how we can identify in the microscope in the slide. Okay, this entire the fibrous part you can identify. Occasionally, here you can see one chondrocyte, and the chondrocytes, they are arranged in the rows, means one by one. Okay, that is a identification of white fibrous cartilage. Is coming to the elastic cartilage. Elastic cartilage identification is very easy. Same pericardium we can identify, and then matrix. So matrix, it is not basophilic as the hyaline cartilage. Okay, it is eosinophilic, and this matrix, it is consisting of chondrocytes. Chondrocytes. Can you see any groups are there? No, there are not any groups. They are individual, and also between these chondrocytes, you can identify small thread-like structures. They are nothing but the elastic fibers. Okay, so presence of elastic fibers in the matrix. Okay, that is eosinophilic matrix, and the pericardium is lining the cartilage. That is the identification of elastic cartilage. Then we are moving to the next one. So coming to the elastic cartilage identification. So here you can identify the chondrocytes, and this is the pericardium, and elastic fibers. These are all the elastic fibers which are present between the chondrocytes. Okay, that is the identification of elastic cartilage. It is very easy slide which you can identify in your practical examination, that is longitudinal section of the compact bone, longitudinal section of compact bone. So whenever you're writing the slide name, be careful, you have to write clear name, longitudinal section of compact bone or transverse section of compact bone, like that I have to write. So which the slide which you're seeing on the screen is nothing but the longitudinal section of the bone. So how to identify that? You can identify the vertical canals. Okay, vertical canals you can identify, they're nothing but the Volkmann's canals. Okay, so the presence of this Volkmann's canals, and these are all called as lacunae and osteocytes, and the lamellae, collagen fiber bundles you can identify. So they're all the key points or identification points of the bone. So here you can see that Volkmann canal, and these are the collagen fibers, they are lamellae, they are called as, and these small gaps, they are called as lacunae. In the gaps, in that lacunae, we can identify the osteocytes are located. Next slide which you are going to see is transverse section of bone. It is also very easily identifiable slide. Okay, so identification points are the same thing. We can identify the osteocytes and the lamellae, circularly arranged lamellae, they're called as concentric lamellae. Then inner circumferential lamellae, outer circumferential lamellae. Okay, so these are all the identification points of the TS of bone or transverse section of the compact bone. Then we are moving to the next one. Yes, here what we are seeing here, which muscle it is. How to identify this is, it is a cardiac muscle. It is a longitudinal section, and this is a transverse section of cardiac muscle. How to identify is, so these are branched. Can you see the branched here, branched structures? So identification point of this cardiac muscle is, the muscle fibers are branched and the centrally placed nucleus and striated muscle fibers. Striated, centrally placed nucleus, and also we can identify the branching pattern of the muscle fibers. They're called as intercalated discs are present. This is the identification points of the cardiac muscle. This is how we can identify the cardiac muscle. So these are the branches, intercalated disc. Okay, so this is the branches of the cardiac muscle fibers. This is called as intercalated disc, and you can identify the nucleus size. Size is bigger and it is present in the center. Okay, centrally placed nucleus and branched intercalated disc is present. So that is identification of cardiac muscle fibers. So next one is skeletal muscle. How to identify the skeletal muscle is, so all the cylindrical muscles, they're all cylindrically present, and the muscle fibers are bundles, and there are multiple nuclei, means multinuclear, and the nuclei are peripherally placed. Can you find these dark structures? These are nothing but the nuclei, and there are many, and they are present peripherally. So this is the identification point of the skeletal muscle. Okay, so that is about the skeletal muscle. Next we are moving to lymph node. So coming to the lymphatic system, how to identify the lymphatic system. So lymphatic system identification is, so this is the lymph node slide which you have to draw in the records also, and also you have to draw in examination also. Okay, so this is called as capsule, outer covering capsule you can identify, in the journals, and lymphoid follicles you can identify in the cortex, and the vessels which you can identify here in the capsule, they are passing through that and enters into the lymph node. They are called as afferent lymphatic vessels. And then at the medulla, we can identify the medullary cords, presence of the medullary cords. And the cortex, we can identify the cortex, we can identify lymphoid follicles. And entire the structure is covered by the capsule. So that is nothing but the identification points of lymph node. And this is how we can identify the lymph node. Actually, panoramic view, we can identify so many lymphatic aggregations, but here you can see that it is a small part of the lymph node. Here you can see the capsule, and here's the invagination of the capsules, and these are nothing but the lymphoid follicles. Okay, so these are the follicles are present in the cortex, and inside it is called as medulla, medullary rays. These are called as medullary rays. Okay, so this is how we can identify the slide of lymph node. Then we are moving to the next lymphatic organ called as hemolymphoid organ called as spleen. So spleen, how to identify the spleen? Same thing here also you can identify the capsule. In invagination of the capsule you can identify trabeculae, and inside that we can identify the lymphatic aggregation, and as well as there is a red pulp and the white pulp. Red pulp means here you can see that in the medullary part you can identify splenic cards, and between the splenic cards you can identify the splenic sinusoids, and this is called as a red pulp. And in this lymphatic aggregation of the cortex you can identify an arteriole that is called as eccentric arteriole, and this lymphatic aggregation is called as white pulp. So presence of the white pulp, presence of the red pulp with eccentric arteriole in the white pulp, that you have to write, and splenic cards and splenic sinusoids are all seen. That is how we have to write in the identification points in the spotters. Yes, coming to the spleen. So this is the identification. You can identify in the microscope in the slide. You can identify like this. So these are nothing but the capsule, and these are nothing but the invagination of the capsule called as trabeculae, and this is nothing but the white pearl. And somewhere here you can see that eccentric arteriole, and this is nothing but the red pulp. Okay, splenic sinusoids and all. So this is how you can identify in the slide of spleen. Then moving to the next organ is palatine tonsil. How to identify the palatine tonsil? This slide, it gives a confusion. Same thing which we can identify in another organ that we are going to discuss in the next session. Okay, so which organ it resembles is, it resembles like the esophagus. Why? Because here also you can identify the crypts. There also in the esophagus also you can identify the crypts. So here in the palatine tonsil you can identify the palatine crypt. Then in the esophagus the esophageal crypt is present. Okay, invaginations, they're called as crypts. And the lining epithelium is stratified squamous non-keratinized epithelium. Same thing which you can identify in the palatine tonsil, and same thing we can identify in the esophagus as well. Then we are moving to the just below to this lamina propria and the submucosa. What are the structures we can identify in the palatine tonsil? Or in the lamina propria we can identify the lymphatic aggregation, lymphatic nodules. Okay, lymphoid follicles are present in the palatine tonsil. There is no lymphoid follicle present in the esophagus. That is the main differentiation between palatine tonsil and the esophagus. Okay, and also you can identify the mucous acini are also seen in the palatine tonsil. So the identification points what you have to write is, the lining epithelium is stratified squamous non-keratinized epithelium. Then the lymphoid follicles are present in the lamina propria, and the mucous acini are seen in this particular palatine tonsil. That is the identification points of palatine tonsil. Yes, this is how we can identify the palatine tonsil. So you can identify the palatine crypt over here, then mucous acini over here, and these are nothing but the lymphatic aggregations, lymphatic nodules. So that is about the identification of palatine tonsil. Next, we are moving to the thymus. So thymus, how to identify is thymic lobules. We can identify different lobules. These are called as thymic lobules. If you observe one lobule, it appears like a, so again inside that lobule we can identify the lymphatic aggregation, and the center of this lymphatic aggregation we can identify some dark pink stained structures. They are called as Hessel's corpuscles. So presence of the Hessel's corpuscles and thymic lobules are seen, and it is covered by the capsule. These are the identification points you have to write in the under the thymus. Now coming to this thymus, how you can identify the thymus, how you can see in the slide. So you can identify these are nothing but the thymic lobules. So and then if you take one lobule at the center part, you can identify some dark pink stained structures. These are nothing but the Hessel's corpuscles. So presence of the Hessel's corpuscles and thymic lobules, these are the identification points of the thymus. Next, we are moving to the nervous system. In the nervous system, we are going to see the two structures, cerebrum and cerebellum. Now, finding the cerebrum, actually, the picture which have projected here is actually a special stain, casual stain. The presence, how to identify this cerebrum is very easy, because all the pyramidal shaped cells we can identify, that itself it is giving you a clue to identify this particular cerebrum. So this is a hand-drawn diagram of cerebrum. You can identify these pyramidal shaped cells are seen, and here you can see that inside molecular layer. Then after that you can identify the granular layer. Then the pyramidal cell layer, small pyramidal cells are present in the external pyramidal cell layer. Then in the internal pyramidal cells you can identify the giant pyramidal cells of beds. So these are all the structures which we can identify in the cerebrum. Okay, in the medullary part, we can identify pyramidal cells are also there present, their extension to the medullary part. Okay, so these are the identification points of cerebrum. How to identify? Simply you have to see clearly that if there are any triangular shaped cells are you are seen in the slide, that is nothing but the cerebrum. Okay, triangular shaped cells. Okay, then we are moving to the next, cerebellum. Cerebellum identification is also very easy. How to identify this cerebellum is, so there are so many numerous folds we can identify. These folds are called as folium. Okay, these folds are called as folium. And then if you observe one particular one fold, we can identify the molecular cell layer above. Then just below that, we can identify basket-shaped cells. Okay, or flask-shaped cells. They are called as Purkinje's cell layer. So identification points, what you have to write is, many folds are present. They are called as folium are present, and Purkinje's cell layer is present. This cell layer which you are observing here. So this is nothing but the Purkinje's cell layer. Purkinje's cell layer is seen. Then under the Purkinje's cell layer, you can identify the granules. That is called as granular layer. So these are the things which you have to write under the cerebellum. So this is a special strained structure of cerebellum. Okay, sometimes these uh, special strained slides are also focused sometimes if HN is strained or not present. Then moving to the next structure is large size artery or elastic artery. How to identify this large size artery is, you can identify a small part of the artery, and also you can identify the tunica media, the middle layer. It is very thicker and it is consisting of abundant amount of elastic fibers. So tunica media, it is consisting of abundant amount of elastic fibers, and proper internal elastic laminae and external elastic laminae are seen. That is how you have to write the identification points under the large size artery or elastic artery. And this is how we can identify the large size artery or elastic artery. So you can identify entire, this is nothing but the tunica media. So you can see these arrows showing the tunica media, and it is consisting of more number of elastic fibers. Can you see these are all nothing but the elastic fibers? So this is giving you a clue to identify the large size artery or elastic artery. Okay, so as a blind mark, you can easily identify that elastic artery easily. The last one which you are going to see is duodenum. Coming to the GIT, the first slide which you are going to see is duodenum. Duodenum is identify the finger-like projections. These finger-like projections are called as villi. So villi, identify the villi. Immediately you just go down and try to see that there are in the submucosa, we can identify so many glands are present. If so many glands are present, that is nothing but they are nothing but the duodenal glands or Brunner glands. So these are all the Brunner glands which are present in the submucosa of duodenum. Okay, so that is the identification of the duodenum. Now moving to the kidney, how to identify the kidney. So identification of the kidney is also very easy. So you try to identify this is a Bowman's capsule which we can identify. I'm going to share another picture which that picture, when you see that picture in the microscope, you can easily identify that. I will show that. Yes, coming to this picture. So this is how you can identify in the slide. Okay, so these are all nothing but the tubules. So here you can see the tubule, this is a tubule. So presence of these tubules, proximal convoluted tubule, distal convoluted tubule. So this is nothing but the Bowman's capsule. Okay, this is nothing but Bowman's capsule and glomerulus, tuft of capillaries. So presence of the proximal convoluted tubule, dark stain, and distal convoluted tubule, that is a light stain, and the presence of Bowman's capsule with the tuft of capillaries. Okay, that is how we have to write the identification points of the kidney. So this is the ureter. Next thing is ureter. So star-shaped lumen you can identify, and it is lined by urothelium or transitional epithelium. Transitional epithelium, already you might have studied about it. That is consisting of the umbrella-shaped cells which are present towards the center, towards the lumen. Okay, these are nothing but the umbrella-shaped cells called as transitional epithelium or urothelium. Okay, so and then below that you can identify the lamina propria consisting of connective tissue, and then muscularis externa consisting of outer circular and inner longitudinal. And sometimes, if the ureter has been taken from the pelvic part of the ureter has been taken and made in a slide, you can identify three layers of the smooth muscle cell layers: outer circular, inner circular, and middle longitudinal smooth muscle cell layer. We can see if the ureter has been taken from the pelvis part and made it a slide. Okay, so that is the main identification of the ureter. Then this is how we can identify the ureter, star-shaped lumen, and you can see this is epithelium called as urothelium, transitional epithelium. Okay, so this is the identification of ureter. Inner circular and outer longitudinal muscle coat you can identify. Next one is urinary bladder. Urinary bladder, same thing, same epithelium we can identify. So many folds we can identify. These are mucosal folds, and it is lined by the transitional epithelium or urothelium. This umbrella-shaped cells, they are consisting of some are uninuclear and some are binuclear. And then we can identify the smooth muscle cell layer, inner longitudinal, middle circular, and outer longitudinal smooth muscle cell layer has been seen in the urinary bladder. Okay, these are the identification points, and this is how you can identify in the histology spotters. Okay, and this is how you can identify the urinary bladder. These are all nothing but the mucosal folds, and it is lined by the urothelium. Then just below to this, you can identify under this is the lamina propria, lined by the connective tissue. Then after that, muscle coat, inner longitudinal, outer longitudinal, middle circular. This is the circular layer, and this is inner longitudinal muscle coat. Okay, and outer longitudinal is present on other side. Okay, so but in the slide only we can identify the two layers of the smooth muscles. Why? Because when you focus that muscle coat, you may miss this epithelium. That's why they focus the epithelium. They focus the epithelium. So according to that epithelium, we can easily identify the urinary bladder.